He is chairman of the European Society of Cardiology Heart Academy and the Council on Cardiovascular Genomics. He is president of Cardiomyopathy UK and executive editor of the European Heart Journal. Professor Elliott is a world expert on cardiomyopathies, and I believe he is the best to bridge the gap between science and clinical medicine. Professor Elliott.
Thank you, Adelina. It's a great pleasure to be here. So, ladies and gentlemen, it's a great pleasure to present to you today at the Hellenic Cardiac Society. I'm really sorry that I can't be there in person, but I think we all live in hope that next year will bring us much more happiness and the opportunity to meet together as friends and colleagues. Today, I will be presenting a short talk on cardiomyopathies in the 21st century, bridging the gap between science and clinical medicine. So it's been customary in my talks to often try to anchor the conversation in a historical reference. And here I'm using a paper from 1957 by Wallace Brigden, who was probably the first person to use this term, cardiomyopathies. His definition was essentially very similar to that we still use in our everyday practice, which is that a cardiomyopathy is a heart muscle disorder which is not explained by coronary disease or abnormal loading conditions as may occur in patients with valve disease or congenital heart disease. I would recommend that you go and read Wallace's paper because it contains a number of very interesting questions, some of which we are still struggling to answer. Perhaps one which may come as a surprise is the actual epidemiology of these conditions. How common are they in our everyday practice? And this statement from his paper in 1957, the true incidence of these diseases is unknown, but is probably higher than is generally supposed, I think applies even now in the modern era. Most of the data that we have come from relatively small or very selected populations, with figures for hypertrophic cardiomyopathy of 1 in 500, dilated cardiomyopathy of 1 to 7 per 100,000, and so on. On the right here, I show you some very recent data from an exercise that we've just conducted in the United Kingdom, looking at electronic health records. And here you can see that the prevalence rates are broadly similar, with 1.5 per 100,000 for ARVC, 4.3 for DCM, 3.5 for HCM. Remember, these are patients within the healthcare system. So this is, if you like, the top of this pyramid of symptomatic patients. The one thing I would emphasize on this slide is that dilated cardiomyopathy would appear to be the more frequent of these subtypes. This slide from the same data set, I think, shows us a very interesting trend that perhaps we've been slow to appreciate. And here you can see the age-related prevalence for women and for men. And while I think we often think of these diseases as disorders of the young, you can see here actually our peak prevalence rates occurring now in the middle to late decades. There's also been a dramatic increase in the reporting of these conditions. And you can see here that the prevalence of cardiomyopathy within the electronic health record has increased by almost 60%. And in the case of ARVC, there has been a dramatic increase in the number of cases being reported in women. So I think this reflects our ability to diagnose, but also greater awareness. I think where we've made much more progress since Wallace Brigden's paper of 1957 is in understanding the etiology, the cause of these diseases. And as he said very wisely at that time, cardiomyopathies may be caused by any of the known disease processes. And he observed then that idiopathic should be diminished as a label for these disorders. And I think that's certainly, as we move into the 21st century, becoming the case. We are finding the cause of disease in the majority of cases of cardiomyopathy. And this is reflected in the most recent ESC classification for cardiomyopathies, where we emphasize this fact that these labels that we use, these phenotypes, describe families of genetic and acquired diseases. I think what we've learned over the past 2 to 3 decades is that the majority of cardiomyopathies can, and perhaps are, genetic in origin. The first paradigm for this came from the discovery of a mutation in a large family with hypertrophic cardiomyopathy by Christine Seidman, John Seidman, and Bill McKenna and others. And this established the paradigm of hypertrophic cardiomyopathy as a disease of the cardiac sarcomere. Following their discovery of a mutation in the gene encoding beta-myosin heavy chain, virtually all of the other proteins that are involved in the cardiac sarcomere were implicated in other families, the next most common gene being cardiac myosin binding protein C. And this story continues to the current age with rare but new mutations being discovered in families, which are slowly chipping away at this unknown section of the etiological pie of hypertrophic cardiomyopathy, if you will. The other thing that we're learning is that within this overall phenotype of hypertrophic cardiomyopathy lie patients with relatively uncommon metabolic disorders, but also patients that have diseases which are not so rare, such as amyloidosis, diseases which hitherto we haven't really been looking for in a systematic way. This breakdown of etiology applies to all of the other cardiomyopathy subtypes, so that the same applies for dilated cardiomyopathy. I'm sure we're all familiar with lists like this that we can read in review articles and textbooks of the many different disorders, insults which can cause DCM. But once again, what we've learned is that many patients, perhaps the majority of patients, have genetic forms of disease. We've known for many decades that if you look at the families of patients with dilated cardiomyopathy, maybe about 25% or more will have a family history. And as we've been able to genotype families, we've started to discover this complex genetic architecture of DCM with genes affecting the cell membrane, the sarcomere, the intermediate filaments, and the nuclear envelope. If we genetically test people with dilated cardiomyopathy, if you have familial disease, then you've got a 1 in 2 chance of identifying a pathogenic mutation. If you have apparently sporadic disease, it remains at 1 in 4 chance. So genetics, as in hypertrophic cardiomyopathy, are equally important in DCM. And the same is true for the third major subtype of cardiomyopathies, arrhythmogenic right ventricular cardiomyopathy, which from the outset we knew was a familial disease. And so ever since the discovery of the first mutation in so-called Naxos disease, and a recessive form of this disease, we've discovered that many patients have mutations in the genes that encode different components of this structure, the desmosome, the structure that anchors adjacent cardiomyocytes together. If we look at mutations in some of the key protein components of this structure, we can account for maybe 30 to 40% of disease. And once again, we continue to find rare but important non-desmosomal genes which can present with a very similar phenotype. So in terms of etiology, I think we have a pretty good understanding of the cause of disease in many, if not the majority of patients. Another theme that we've seen in cardiomyopathy over the past 50 years or so is this improvement in our ability to diagnose these conditions. And this has reflected really changes in technology. So we've moved from the stethoscope and physical examination through ventriculography, ultrasound using M mode, 2D echocardiography, and obviously in the modern era, cardiac MRI. There has been in the recent literature and in the current literature an increasing focus on our ability to, if you like, characterize the underlying myocardial substrate. But there are also some semi-quantitative approaches to modern imaging, which also provide us with key information. So just looking at the presence and pattern of scar can identify subsets of patients with dilated cardiomyopathy, for example, that have genetic disease. We've also, interestingly, gone back to the future, if you will, and we're rediscovering nuclear medicine, not for its traditional use in looking at myocardial perfusion, but in actually making specific diagnoses. Here, the use of bone scintigraphy, for example, in diagnosing TTR amyloidosis. And I think we're also now rediscovering the entity of myocarditis by using non-invasive imaging, as in this example, using positron emission tomography. But relevant to this talk, perhaps the biggest improvement we've had in diagnostics is our ability to perform genotyping in the clinic. Once again, this reflects changes in technology, which have allowed us to do high volume, relatively low cost genotyping. And we can now imagine in the not too distant future, the ability to maybe do this almost in real time, so that the ability to genotype will become as easy as doing a full blood count. I think the question which I was asked to address here, which is, how do we bring together science and clinical medicine in the 21st century, really builds on this knowledge. And in particular, I think we're now entering a new and exciting therapeutic era in cardiomyopathies. I think if you stand back and look at the generality of cardiovascular medicine, we've had several decades of incredible success in reducing the mortality from heart disease, whether that be atherosclerosis or heart failure or hypertension. Here you see an example of the impact of drug trials on sudden death rates in heart failure populations. And I think this has given us a sense that actually, we're really reducing the unmet need in cardiovascular medicine generally. To some extent, this is true of cardiomyopathies as well. So here, if you look at the survival rates from the 1970s, 80s, 90s, and the 2000s, in patients with dilated cardiomyopathy, this too has improved dramatically, reflecting again the power of modern pharmacotherapy of heart failure. But I think what we've learned in the cardiomyopathy field is that within this overall picture, there lie subpopulations in whom there is still much work to be done. In this nice study here from Luisa Mestroni's group, looking at patients with dilated cardiomyopathy, you can see here that about 30 to 40% had a genetic variant. If you compared the outcomes in this case for sudden death or ventricular arrhythmia, then those who carried a variant clearly had a poorer prognosis than those who didn't. But even within that group, there were clearly genetic variants that carried a much, much higher risk. In this case, patients who had variants in desmosomal genes or in the lamin AC gene. And it's these data that are hidden within these large population studies, heart failure trials. The same is true for other cardiomyopathies. So in hypertrophic cardiomyopathy, overall event rates now are very low. So we're looking at maybe 1 to 2% per year for all-cause mortality. However, there is still a small excess mortality in hypertrophic cardiomyopathy throughout the decades. And what we're seeing, of course, is that as patients live longer, we prevent sudden cardiac death, for example, very successfully. We're starting to see patients who develop progressive systolic impairment. Now, this shouldn't really come as a surprise. This is a myocardial disease, which is progressive, but it takes decades to progress. And we see loss of cardiomyocytes, progressive fibrosis, which ultimately results in adverse remodeling and the development of heart failure. If you look here from that same study that I showed you, at the cause of death in patients with hypertrophic cardiomyopathy, the dark bars here are sudden death, and you can see as patients age, the incidence of sudden death reduces. But in the gray bar here, at every decade, there is a significant proportion of patients dying from heart failure. Once again, just to drive home this message, this time from the SHARE registry, a predominantly North American registry, if you look at what happens to patients over time with hypertrophic cardiomyopathy, you can see that many, if not the majority, will develop heart failure, particularly if they have early onset disease. So within dilated cardiomyopathy, we have patients who have a higher incidence of sudden cardiac death and maybe heart failure progression despite modern therapy. And within hypertrophic cardiomyopathy, we have this insidious but continuous development of heart failure over time. And that leads me to this concept of so-called precision or personalized medicine. Because what I'm talking about here is really making a definitive diagnosis, understanding the etiology of disease so that we can then develop more bespoke, more precise, if you like, therapies for that particular subgroup of patients. Even a casual reading of the current medical literature will show you that there is enormous interest in this area, with a huge number of technologies and sciences being developed to try to promote precision medicine. And this can, I think, sometimes seem a little overwhelming. Essentially, however, I think a lot of this reflects, again, our technology. So, for example, we can now perform not only genomics, but transcriptomics, proteomics, metabolomics at scale and very rapidly to better understand the pathophysiology of disease. With the power of modern computing, we can examine large data sets and try to identify disease clusters within those data sets. I think we've now moved into a much more collaborative network-based model of research, both nationally and internationally, which again allows us to increase the power of our scientific studies. And of course, we're developing methods to gather data in new ways with wearable technologies and so on. Now, this approach of ever more granular definition of disease is being used in some areas of medicine very effectively, in particular oncology, where sort of existing, more conventional disease phenotypes, breast cancer, lung cancer, etc., are being broken down into new clusters, new groups built around a more detailed understanding of their pathophysiology. In cardiovascular medicine, as this article in Nature observed, we're perhaps a little bit behind the ball on this one. As you can see here, the statement was that personalized medicine has yet to make much headway in the treatment of cardiovascular disease. And I think there are many reasons for that. There are some aspects of cardiovascular medicine which make it harder, for example, getting tissue to make a diagnosis. I think we already have a paradigm which has been very successful, and perhaps there hasn't been seen to be a need for this more granular approach. But I think the big thing that we have to do is to show that by making the effort to make it etiological diagnosis, that has an impact on outcomes. And I think that's really the challenge and the opportunity that we now have in the modern era. Now, this notion of genetic guided therapy might seem a little bit science fiction for cardiovascular medicine, but we are to some extent already doing this, particularly in the inherited diseases. So, for example, if you think about inherited channelopathies, we already make clinical decisions based upon a genotype, whether it be long QT syndrome in choosing the drug or deciding whether to put a defibrillator, lifestyle adaptation, the same with CPVT or Brugada syndrome. Increasingly, I think we recognize that in the familial aortic syndromes, the threshold for surgical intervention in patients with aortic aneurysms varies according to the gene, because we realize that some genetic subtypes are much more likely to develop aortic dissection or rupture at a smaller aortic dimension than others. We've recognized within our populations of cardiomyopathy that there are rarer diseases, but for which there are specific therapies. So, for example, Anderson-Fabry disease in patients with hypertrophic cardiomyopathy, for which there are enzyme replacement options. Familial hypercholesterolemia, perhaps something that many general cardiologists now encounter. And here we're looking at therapies designed to interrupt the underlying biology of this disorder with PCSK9 inhibitors and soon genetic therapies to try to knock down PCSK9 more effectively. I think one of the most recent discoveries, which has really given a stimulus to the field of cardiomyopathy, has been our focus of attention on amyloidosis. You'll recall that amyloidosis affecting the heart occurs in two main types. There's that associated with plasma cell dyscrasia, which results in the deposition of light chain fragments as amyloid fibrils. And then the more common type, which is caused by deposition of fragments of this transport protein, transthyretin, produced by the liver, again as fibrils within the heart. And this can occur as a genetic form or a more common acquired form in older age. There is now intense interest on this family of diseases with various therapeutic pipelines being examined. And one of these is looking at drugs which can stabilize transthyretin. Transthyretin is a tetramer which in amyloidosis dissociates, and the fragments then reassemble as amyloid fibrils. So there are now drugs available which stabilize this tetramer and prevent, theoretically, the deposition of amyloid fibers. And we've recently had this study in 2018 of one of these agents, tafamidis, a TTR stabilizer, which has shown a 30% reduction in mortality and stabilization of quality of life and exercise tolerance. So we're now seeing other therapeutic options which are being explored in the cardiac space, this time using genetic therapies to knock down production of TTR within the liver using silencing RNA technologies or antisense oligonucleotides. And there is a signal already in some studies showing that there is not only stabilization, but the prospect for perhaps reversal of some of the features of these diseases. Now that's a very specific disorder, but the same approach is now being pursued in some of the structural genetic abnormalities that we see in DCM. So if we go to laminopathies, maybe 5, maybe 8% of dilated cardiomyopathy. Lamin is this protein which sits just underneath the nuclear envelope. We understand its phenotype very well. It starts with AV block, atrial arrhythmias, progression to sudden cardiac death, even with only very mild impairment of LV function, and then slow progression to more advanced heart failure. We've recognized that this is a group with a high risk of sudden cardiac death, and this has already made its way into our society guidelines, where we recommend defibrillators in patients with laminopathy. But by focusing on the underlying biology of this disease, we're also now developing new therapies which can potentially treat the underlying disease itself. This is a complex slide, but what it's showing you is that lamin sits at this very important signaling interface between the extracellular space, the external environment to the cell, and gene transcription. And there are a variety of pathways that transmit external stresses or signals to the nucleus, which are vitally dependent on this regulatory role of lamin in the nuclear envelope. One of the pathways which is overactive within patients who have lamin mutations is this pathway, the P38 signaling pathway. And there are now inhibitors of this pathway that are in trials, which in phase 2 studies at least have shown improvements in exercise tolerance, reductions in BNP, and improvements in ejection fraction. In hypertrophic cardiomyopathy, the story is the same. So we've gone back to the biology of this disease to try to understand the impact of mutations on this key function, the interaction between beta-myosin, heavy chain actin, and the regulation of contraction at a cellular level. And we've focused particularly on the beta-myosin heavy chain in this instance, where we know that some mutations result in a hypercontractile phenotype. Other mutations result in a hypocontractile phenotype, and this results in hypertrophic cardiomyopathy and in dilated cardiomyopathy. Drugs are now being developed which try to correct this abnormality. So this rather extraordinary slide you can see here, which is looking at a compound which is now called mavacamten. What you see here are actin filaments moving across a sheet of myosin within a well. And you can see here normal on the left, and here inhibition of this interaction between actin and myosin with the drug mavacamten. This drug has created again some recent excitement because it has now been shown in a randomized trial published recently in The Lancet to treat one of the manifestations of hypertrophic cardiomyopathy, outflow tract obstruction, reducing the gradient, almost abolishing it in a third of patients, and resulting in improvement in exercise tolerance, reductions in BNP and troponin. And now similar drugs are entering trials for DCM. So rather than inhibiting actin-myosin interaction, drugs which enhance it. So what you're seeing there are some of the exciting examples, if you like, small molecule therapies which are designed to modify the pathophysiological abnormalities in disease. But even more exciting is the move now into gene therapy, something which is a reality in myopathies, hemophilia, metabolic diseases, but which is now being contemplated in patients with genetic forms of cardiomyopathy. Trying to correct the underlying gene defect by introducing wild-type gene through a variety of methods. One which is under active investigation is this use of AAV, adenoviral vectors, to deliver the wild-type gene to the heart. We know that in animal models this can be done and has an effect. Here you see work from the Hamburg group, Lucy Carrier and Thomas Eschenhagen, where you can see here that by using this vector approach to deliver wild-type gene, you can increase the amount of wild-type protein within, in this case, pluripotent stem cells, and you can also reduce the cell size, showing that you get normalization of the organization of the sarcomere within the cell. The company is now investing in this space, in hypertrophic cardiomyopathy, in dilated cardiomyopathy, and also in ARVC. So I think as we look into the 21st century, we can start to see how we're going to move from this effective but quite static range of therapies we've had for the past couple of decades in cardiovascular medicine to an even broader range of therapies, including gene delivery, small molecules, silencing technologies, something I don't have time to get into today, but immunotherapies, which are already showing promise in atherosclerosis. And of course, may have enormous promise in inflammatory diseases, myocarditis. I think this scientific frontier also allows us to imagine a future where we don't only treat patients that have established disease, but where we could even prevent disease from developing. Now, these are data from an animal model of hypertrophic cardiomyopathy administered with mavacamten, and the phenotype was arrested if the drug was given early enough. Now, we've been here before, but this just gives us a tantalizing glimpse of where we may be in 10 or 20 years' time, where we make a genetic diagnosis and give a therapy to prevent disease progression. So that's a bit of a whistle-stop tour through cardiomyopathies. I hope what I've been able to convey is the sense of, I think, enthusiasm, excitement we now have in this field, where we've now moved, as I say, into the therapeutic era. We have a whole range of different options, which even if only 10% succeed, will allow us to impact on the quality of life and the prognosis of patients with cardiomyopathy. Thank you very much for listening.